Okay, so this is continuing with our talk about biotechnology. Last time we talked about how we would get genes of interest that we wanted to work with into bacteria. Why would we do that? Well, that goes back to this slide here. Um, uh, if we're going to do anything with a DNA sample, it generally has to be big enough to work with. And bacteria, if you'll remember, we talked about uh, DNA molecules being inserted into little genes being inserted into little DNA molecules in bacteria that we call plasmids. There's a main DNA molecule, but there are all these little accessory molecules that we call plasmids. But the thing is, once we get that into the bacterium, then that bacterium, every time it reproduces its DNA, it reproduces its plasmids. So when one bacteria becomes two, and two become four, and four become eight, and eight becomes 16, and 16 become 32, and so on, every time that happens, we are making new plasmids. So we are making copies of the DNA we wanted. We are making copies of that DNA we wanted, and additionally, if that DNA is working, it is making protein. So if what we really wanted is the protein, maybe that human insulin we were hoping to give to someone who might be diabetic, it's making lots of that too. So that's one reason bacteria have been the workhorses for DNA biotechnology for so long, is because once they get the DNA in them, they just start copying it on their own and making tons of the protein that DNA codes for. Okay, so let us talk now about some of the other tools that we need. We talked about cutting DNA. We've got to cut out the gene we want. We've got to cut open a plasmid, for example. So let's talk about the tools that allow us to do that. Okay, very seldom is a whole intact chromosome very useful in biotechnology. What you want usually is a particular gene. To study or to analyze, whatever. So it's the specific genes that we actually use. So how can we actually cut a DNA molecule? We can figure scissors probably are not going to work for us in this case. Well, what will work for us are enzymes. There are enzymes naturally made by bacteria, actually, that whose job it is to actually cut DNA into pieces. Okay? These enzymes are called restriction enzymes, and scientists, human beings, have learned to harness these restriction enzymes that are made by bacteria and to use them as we need them to cut DNA into pieces. Another name for these restriction enzymes is restriction endonucleases. So if you see that, you can just think that. Notice the ace means it's an enzyme. Okay? They've been around for a while now and do understand their origin. They actually evolved in bacteria, and the reason bacteria have them is it's kind of a self-defense. Bacteria use them to cut up foreign DNA that might get into their cells, usually from a virus. Bacteria get viruses too, and so this is one of their defenses against virus DNA taking them over. Okay, so protection against, okay. We don't really need to worry too much about that. This bottom line here is kind of answering the question, how is it that these restriction enzymes don't cut up the bacteria's own DNA if they have DNA's DNA, right? Well, it works very well because bacteria just make sure that they don't have any of the particular genetic sequences that these restriction enzymes are going to be recognizing. So let's talk about that. How do restriction enzymes work? Do they just randomly hack up DNA? And the answer to that is like sort of, but they only randomly hack it up at very specific, predictable locations along the DNA molecule. And the places where they do this are called restriction sites, and you need to know that. And what makes a restriction site is a very specific sequence of DNA nucleotides or letters A, T, C, G on the DNA strand that the restriction enzymes will recognize. And again, the bacteria is going to make darn sure it doesn't have any restriction sites in its own DNA, but viruses may well have those sites, and then they will be cut up and destroyed. So, let's look at these. What do you notice about these phrases? Notice anything? Radar, race car, madam, I'm Adam. These phrases are all palindromes. They are phrases that are spelled the same backward as they are forward. Okay? It is sequences like these that are recognized by restriction enzymes. They are palindromes of A's, T's, C's, and G's. Okay, so here's how they work. The restriction enzymes are looking for a symmetrical palindrome. So something like this, actually outlined in the red here, GAATTC, and over on the other side, GAATTC. That's a symmetrical palindrome, same forward as backward. Now, let's say a restriction enzyme recognizes this palindrome and it will cut it. And sometimes they cut in a way that when these two pieces are separated, there will be exposed bases here and exposed bases here. Those are called sticky ends, meaning if we get I'm just going to kind of draw the geometric shapes of what would be separated. Okay, so like this CTTA would be CTTA here. This would be ready to bond with its opposing nucleotides, as would this piece here. Maybe it's AATTC here. It would be ready to bond with the opposing nucleotides. They're sticky, they're exposed, they're ready to bond. Okay, so they will bind to any complementary DNA that they find. So that's really helpful when you're trying to stick DNA from one source into another. If they've got the complementary sticky ends to their DNA, then they'll stick right together very readily. Now, some restriction enzymes don't work that way. Some just go chop right down the middle, and if we do that, then we have what are called blunt ends and nothing can stick. So those aren't particularly useful. It's the sticky ends that we want if we're doing genetic engineering. Now, there are many types of restriction enzymes, and they all cut at a slightly different palindrome, so they'll recognize different restriction sites, and they have been named after the organism in which they were found. EcoRI, for example, is E. coli, restriction enzyme 1, Hindi3, I can't remember what the name for that, but they're all different just because of the different bacteria in which they were discovered, and the 1s, 2s, 3s are the order in which they were discovered. All right. I only tell you that because we'll be using them in the lab, and so that's what the names refer to. Now, so we've cut some DNA up with restriction enzymes. What in the world can we do with those pieces that we've cut apart? Well, as we've already discussed in our talk about bacteria, we can take a gene from one organism and put it into the genome of another. Okay? If we cut the gene out over here and stick it in over here, those sticky ends might allow us to make a new piece of DNA. We may do that because we want to give an organism some desirable traits. We want a tomato to not spoil so quickly, for example. Maybe we want to cut up DNA to determine the nucleotide sequence of a gene, and when we cut DNA up into fragments, we can analyze those fragments and determine a lot about the genes in the DNA. We can determine actually the orders of A's, T's, C's, and G's. Okay? We can compare DNA from different people when we have cut up a DNA sample into many pieces. And why would we do that? Well, if we have a crime scene, we might want to compare DNA of different people to figure out who might have done the crime. We might want to use it for medical diagnostics. If we think someone might have a genetic disorder, if we can identify the gene, we can give them a genetic test and determine if they have the gene or not. We can use this kind of analysis to determine paternity. If somebody is trying to say they are not the father of a child when they are, we can pretty conclusively determine that. We can also not just compare DNA from different people, but we can compare it from different species. And when we do that, we are actually determining evolutionary relationships among those species, which tells us how the tree of life might actually be assembled. Okay? So let's talk about comparing DNA from different people. And what we would have to do if we wanted to do that. One common way to compare DNA of different individuals is to first cut it into pieces, and then secondly, separate those pieces according to how big they are. If you randomly cut a long DNA molecule, so here's my long DNA molecule, if my restriction enzyme cuts here and there and here, then I'm going to end up with fragments that are of different lengths. And I can use that information to my advantage, okay? But I've got to be able to separate those pieces and be able to determine how long they are. And the way we would do this is by using a technique called gel electrophoresis, okay? Gel electrophoresis works like this, and we will be doing this in the lab, okay? You will get a slab of gelatin. It's a lot like Jell-O. It's agarose gelatin. It's lab grade. It's reasonably pure. And you will place little depressions, little holes. They don't go all the way through the gel, but little depressions in one end of it, and those are being highlighted here at this end of the gel. Okay? And then, if you've got DNA that you have cut up using restriction enzymes, you've got some samples in this files. Maybe this is individual 1, individual 2, and individual 3, and you want to look at those. You just dump the DNA into those little depressions at the end, and then you put the whole thing, so I'm going to put my DNA into here, and into here, and then you put the whole gel into an electrical field. Okay? Why? Okay? DNA actually has a negative charge. It pretty much has to do with those negative phosphate groups that are part of the nucleotides. If you put the DNA in an electrical field near that negative cathode, near the negative pole of the field, if you will, and put the positive pole at the other end, the DNA, being negative, is going to be attracted to the positive, and it's going to be pulled through that gelatin. Okay? That gelatin is porous, and its pores are pretty tiny, so it's acting as a strainer. The small pieces, the small fragments that we cut up in these test tubes, can kind of zip right on through pretty quickly. But the big pieces get held up because they can't get through the holes as fast because they're too big. Okay? So the larger pieces move more slowly, and thus the fragments are going to get separated according to size. Okay? And if these are actually DNA molecules from 3 different individuals, since no 3 individuals have identical DNA, we are likely to get, unless they're identical twins, we are likely to get different banding patterns in the DNA gel that if we stain the DNA, we can see them, because everyone's DNA fragments will be of slightly different lengths. So let's look at this, okay? So there's our negatively charged, that's the source of our negative charge right there, and when we put this DNA in an electrical field, there's the negative end here, here's the positive end, and we are looking at the gel like we're looking at it edge on. So right there, that brown is the whole sample of DNA. We've already cut it using restriction enzymes, so there's many different fragment sizes in there. It's very tiny, we can't see it, but if we turn on our electrical field, okay, the DNA the fragments are going to be pulled through at different speeds. The smallest fragments will go very quickly towards the end of the gel, whereas these bigger fragments have a harder time making it through the pores, and they get held up back here. And so what you end up getting is a banding pattern. If we look at the gel from above now, as we generally look at it, so here is our little depression or our well we started with. So the littlest piece went through and got way down here. The next piece got almost that far. The next piece was slower, and that really long piece got held up way back up here. And so our banding pattern would look like that. Maybe a different individual didn't get as many fragments from the same restriction enzyme cutting up the DNA. Maybe a different individual, we put their DNA in, and we only get a banding pattern that looks like that. Or maybe we get an individual that got the 4 fragments, but they're of different lengths. Maybe there's 2 that are here and 1 that's here and 1 that's way back here, or something like that. So, because different individuals have different DNA sequences, we get different banding patterns, and we can tell individuals apart. Okay, this is just showing you again what we just talked about. We'd get a gel that looks something like this. If we have 5 samples of DNA, they'd have different banding patterns that would result from different individuals. Okay. We would stain the DNA as well, because otherwise it's kind of colorless, so we would stain the DNA. The stain binds to the DNA and allows us to simply visualize it. Many stains also help to weigh the DNA down so it doesn't float out of the little well that you've put it in. Okay? I think maybe we will stop there and pick this up with an explanation of why this works, which we've just talked about a little bit, but we'll get a bit more of a concrete picture of it. Okay? Thanks for watching, and I'll catch you in the next podcast. Take care.